exam and they're going to go off and take it, and the rest of you can get your exam back. And I will just tell you one thing we're going to try to do now is record each lecture and put it on eCampus. It takes, what, 2 hours to get it to download one of these?
This one, it shouldn't be that way because it's right. It's directly on the.
Oh, okay. We're hoping it doesn't take this long. And so I know it's not, the resolution really isn't as great. Those are, I know, are 2 gig from before. But there are a couple of thoughts. I know some of you aren't going to be that happy with your grade, and this will give you a chance to go back and rewatch the lectures and to take more notes and to make sure you're hitting the big parts. I think that'll help everybody. And if you're going to be gone, then at least the lecture's there, and it helps you because I know that we have some times that we're gone. I think I'm only going to be gone 3 more times this semester. And so again, I'll record those lectures and put them on. The 4th time I'm gone is October 30th, and you'll be taking an exam that day. So Kelly, we did schedule it. Kelly will be your test monitor again. I appreciate that, Kelly. No problem. But, and trying to look at what would help you all, I think that would be beneficial. And also then, I don't have to ask somebody to lecture and make sure that they're emphasizing the same things I am and get them to do the test. And as I told you, I'm a little selfish about this class. It's my favorite. So I do want you to know, don't be discouraged with your exam grade. I've heard you are a good class. I know you're coming to class. I know that you're trying. And the first exam is always the hardest because it has the most memorization on it. And also you're getting used to taking one of my tests and what level that I expect. But I will tell you that I graded the test yesterday when I was in, I was called for jury duty. Luckily, I'm here today and not in a jury because I did not want to be with that particular case. And so it does help me. I grade them 1 question at a time. So like question 2, I grade all the question 2s. And I fill out my key and then I grade it, and then I actually do think about what information I got back to you and what information I didn't communicate well. I'll make adjustments, hopefully in my lectures. That's my intent, to do that and to make the class better each time. And I also get to see what concepts you struggled with the most. And I can really tell what things you know and what things you don't know, and I try to give partial points. But I have the key up here so you can see where I gave points, even though these guys have to leave the room when we get to that point. And I would love to discuss with you any of the issues you have about the test. Don't feel like, well, I can't go in and talk to her. So, ready to go for today? So, we're on the next part, on the physical factors affecting meat quality. So, we've talked a lot about all the physical components in there. We talked about collagen and lipids and muscle. Blood and muscle, right? Ultrastructure, how it works, how that muscle contracts, where it gets its energy, how it goes through rigor mortis. A lot of information, isn't there? And of course, there's some more depth we could go into in each of those topics, and that's what Dr. Smith goes into in his class and other classes that you can take. But hopefully I've given you a good foundation. Well, as you know, most of you know, I'm an applied meat scientist. I measure some of the components in the lab, but most of my research relates to how meat is used by consumers. So I'm very interested in the physical factors and how they affect meat palatability. So, well, what I did in putting this lecture together, which I did a number of years ago, and you can see I took different parts of the book, didn't I? I mean, this isn't, the book doesn't have a chapter on this, but as I taught this material, it became apparent that I needed to help students put together how these factors work together. So what we're going to talk about, we're going to talk about the 4 physical or structural components that affect meat quality. And then we're going to talk about meat aging, that resolution of rigor. We're going to talk about that now. And then we're going to talk about cold shortening and the role of electrical stimulation. So we're going to bring in some of the rigor components. And then heat shortening and thaw rigor. We're going to talk about marbling and marbling theories, and then the role of connective tissue. So I'm kind of taking all the components and putting them more on a practical basis. So there are lots of things here, and there are lots of articles, aren't there? And we'll talk about how we want to use the articles for the next class here. So one of the things that I, as a researcher, that I would get frustrated with is, and it's the reason you have so many articles here. I can read Kamari and talk about resolution of rigor. I can read Smith et al. 1987 and all the work on marbling and look at how each of those components affect meat quality, meat tenderness, meat flavor. But every time you do that, you're just looking at a piece of a puzzle. And so I put together this concept, and this is an old Simmental steer. It's supposed to be their ideal from the 80s, so I like this steer. So anyway, so I thought about it. I'm like, you know, as we look at factors that affect meat quality, we know resolution of rigor or muscle fiber degradation influences it. Remember I told you the story about McCormick versus Kamari at the ICOMS meeting, right? So what Kamari was saying is, no, this is the only piece that plays a role, right? And Richard had just, and actually it was during that talk that I put this concept together. But Richard was talking about how the connective tissue can sometimes play a role. And we know that muscle fiber contractile state plays a role, how an animal goes through rigor, and the rigor components that affect it. But we also know that intramuscular fat affected. And did I tell you the story about the first time I went to Brazil to talk? And I'm standing up there, and I'm giving my talk, and they have a translator going from English to Portuguese. And it was luckily simultaneous. I'm talking away, and I think I've just done a wonderful job of presenting my information. And it's time for questions and answers. And I knew there's a gentleman that most of us in the U.S. know, and he stood up and he asked me a question, and it was about marbling, because that was part of what my talk was about. And I answered him, and then somebody else over here stood up, and the two of them started arguing in Portuguese. And the translator stopped translating because it was quite, and it was like two sides of the room, and they're having this argument, and it became very, and I'm sitting there going, well, I wonder if I did what the people who invited me and came my way wanted me to get shipped out of the country. What they were having a discussion about marbling. But when you back up, yeah, it's fine to look at each piece of the palatability puzzle. The problem is that each piece doesn't account for large proportions of variation. So if we look at the animal as 100% of variation, we know that marbling only accounts for 10 to 20% of the variation in eating quality. And in certain situations, muscle fiber degradation only accounts for about the same amount. Now I can create situations where I can change that a little bit. I can give you prime and standard, really high marbling and really low marbling, and I'll account for more variation. But when we're talking about the general population, we're going to just concentrate on beef today, because I'm going to come back with pork quality in a subsequent lecture. And some of it affects pork as well, but not as much. They have some other issues. And so I can take each piece of the palatability puzzle, and I can show how it affects tenderness. And we in an industry do things, that's what I'm going to talk about in this lecture, to try to make each piece not be a negative piece. But each animal has its own palatability puzzle. For some animals, connective tissue might be a driving factor. Older animals, right? Animals, bulls, we'll talk about that. Animals that haven't been fed, we'll talk about that. But that piece of the puzzle may be a driving factor for that animal, but for a different animal, maybe it's contractile state. So the concept here is that every animal is unique. It's called individual animal variation. I call it job security. Because the other thing is I drew a line here, and notice this big part, and actually this line should be up like right here. And that's called unknown. That's called unexplained variation. That stuff in statistics that you hate to hear. And probably most of you go, oh, she's giving a statistics talk. No, not this time, Thursday. That's Thursday. Okay. So this is actually about 80% of the variation. We really don't know all, we can't always explain palatability or eating quality. And we're going to define eating quality as tenderness, juiciness, and flavor, right? Because that's what consumers consider it. That's what we measure as meat scientists, whether as a trained panel or with consumers. And most of this was developed off of tenderness. So the other thing about the pieces is that I made this a puzzle because I like puzzles. I'm a scientist. I like to solve puzzles. And so, and I made them like, you know, very smooth edges and with a black line. It's not quite that black of a line and that defined of a piece, all right? There's interaction between these pieces, which I hadn't ever figured out how to do that. So this is a complicated picture. And what we're going to do in this series, this lecture, is talk about each one of these pieces of the puzzle and how they individually contribute. But remember, when you go out into the large population, I'm going to talk about a study we did where we measured all 4 pieces of the puzzle in a very good population of animals representing cattle in Texas and Louisiana and Florida and the South. And you know what? We only account for 10 to 20% of the variation in tenderness, measuring all the pieces of the puzzle. And for some animals, one piece was bigger than the other. Maybe it was a marbling piece, or maybe it was a muscle fiber degradation piece. So we're moving up in complexity in the way that you think about these. So what I want to do now is I want to talk about each piece of the puzzle. So let's talk about the first piece. And those pieces were not put on there in a certain order or anything. It just have to be whatever I put on the slide, right? They're randomly located. But let's talk about resolution of rigor or meat aging. So I'm going to go ahead and put that up on the board, meat aging. And the story that I told you about Kamari, that's what Kamari was talking about, right? He was talking about resolution of rigor and how we get some improvement in elasticity, right? The muscles open up a little bit and we get some elasticity back. And it's that 4th stage. Well, resolution of rigor, up here I put, everything begins to fall apart. And there's a saying that in Christian religion about ashes to ashes, right? Everything falls apart. Everything's going to degrade. So in meat science, we consume it before it gets to that point, right? We hope. Anyway, so we know that the structural components are going to start to fall apart. And I like to look at this like a house, like you've just built a house. And when you first build a house, you have very strong structural components, right? Structural walls. Those are the structural proteins that you learned about in ultrastructure. The Z-line, Desmond, Nebulin, Titan, all those. Remember all those that you had to know for the exam, right? So everything's nice and, ooh, everything's put together. But with time, there are going to be changes. And you're going to see degradation. And think about an old dilapidated house you might drive by where the roof is sagging and you see things starting to fall apart. That's basically what happens inside a muscle where we see structural components start to degrade. And so when we look at physiological changes, and one of your articles I gave you is Kamari, 1988. I did give you Kamari 88, didn't I? Did I take it out? Just a minute, I'm looking. Oh. I gave you Kamari and Giesing. Okay, Kamari and Giesing talks about this as well. So this is in one of your papers, and it's repeated a couple places. So one of the things that we know that happens, And if you look in a light microscope and you look at muscle immediately post-mortem, and then you look at it 14 days later, 28 days later, what you see is you see that the Z lines are degrading. You also see that troponin T is degraded. And actually this appearance of the 30,000 Dalton subunit, and that's just a subunit of a protein. Right? So when we start seeing smaller fragments, that's an indication that some subunits coming off of some protein. And we do this with gel electrophoresis. So we see that early on we see Z lines, we see troponins all intact, the 3 units of troponin, right? We see nebulin there underneath the thin filaments holding it up. We see titin holding up those thick filaments, and we see desmin connecting the Z lines. But as we watch that muscle degrade, we start losing Z line definition. The Z lines actually, we see fragmentation there. Troponin T is degrading, it's not there, and we start seeing this smaller protein appear, which is most likely the subunit from troponin T. Nebulin, titin, and desmin are not as organized, and they're not there. So it's like they're falling apart. But what's interesting is that actin and myosin don't degrade. So if we're looking at a muscle, and we see early post-mortem, right? We see actin and myosin, they're going to be attached here, right? You know how I draw. This is so you use your imagination, right? Think about it, keeps you awake. Okay. But we start seeing with time that the main structural components, nebulin, titin, and desmin, boy, those are big proteins that really hold everything together. And then the Z line starts to disappear. So we start seeing the Z line disappear. We start seeing that this active myosin component, it stays, the thin and the thick filaments, they stay bound to each other, but we start seeing fragments of them come out because titin, nebulin, and desmin aren't there. We see between myofibrils and myofilaments, we start seeing them loosen their structure because those structural components are there. There's actually a new theory about actin-myosin interactions. There is some thought, and I gave you the Gall et al. 1996 paper, or 1995 paper, that was presented at ICOMS in San Antonio. And what Gall says in that paper is that, yeah, you know, we have, here's a thin filament, right? Here's a head, here's a thick filament. What Gall is saying is, we never see actin and myosin come apart. And we can put trypsin in there and make them fall apart, but that's an artificial enzyme. We have to do that. But during all this falling apart, we see the thin and the thick filament. This is supposed to be a thick, that's supposed to be a thin, okay? Stay together. But what Gall said is that we think of that as just, okay, myosin heads went up, right? We get 100% of them binding, they contract, and so in the early stages, here's a thin filament. Let me do these in different colors, maybe that will help my drawing a little bit, okay? We'll see the heads like that, and they're very stiff. So this is early. And that's, you know, grab, they pull, they're there, they're tough, right? But with time, and you're going to read the Gall paper, and it talks about those first three things up there. And then it starts talking about cyclic AMP and ATP degradation, and you're going to go, she wants us to read this. Oh my goodness. I do want you to read it. I don't want you to worry that much about some of those details. I think some of it you'll absorb. But the reason I want you to read it is it was a new thought And there's also crystallography, which is a new method of looking at things and a new technology at that time. And what Gall said is that with time, they found that the strength of this myosin head, so it's really in the neck region, that with time that weakens. And that there are really kind of, you know, as scientists we know it isn't always one or two phases, but it's a gradual process. But what happens is that we go to, here's the thin filament, here's the thick filament, and the head slowly relaxes. And what I like to think about is that, you know, if you are a weightlifter and you're lifting up heavy weights, you know, at first you have all the, everything, but if you had to hold it there, you'd slowly lose some of your strength, wouldn't you? And so this weakens right in here. And so it relaxes. See the difference from there to there? I purposely drew that. That's not my bad drawing. I purposely drew that to be more relaxed. And so you can see that he presented in that paper, the Gall et al. 1995 paper, he presented the thought that, oh, there is something going on here with actin and myosin, even though they don't degrade, which is what we normally would think about happening to make them more tender. But we see the actin-myosin interaction loosen. It loosens. It isn't as strong. And that happens with time. And so it's going to be that how strong that, or how rigid that head is, and it has to do with ATP degradation, and that's why cyclic AMP is involved in that. Okay. We're not going to get into that much detail. I'm trying to present some papers that have some new thoughts. There's some other things in that paper I want to bring out too. But that's the new theory, that there is a weakening of the myosin heads with time. And while actin and myosin stay bound to each other, that if we were going to break through it, this is going to be the toughest, and this is going to be more tender. Now, is that change huge? Does this make it so that this is like a tenderloin, and this is really, really tough? No, it's a subtle change, all right? So this is, I'm going to just put subtle change. But it's probably happening at the same time that we see Z lines disappear, troponin T, nebulin, titin, and desmin start to degrade. We see all those major proteins degrade. So what that tells me is that everything's starting to fall apart. It isn't just the structural proteins, but we see a relaxing of those contractile proteins as well. So that's what's happening in resolution of rigor. And this picture is to show you that. So this is immediately post-mortem. You've seen these pictures, this picture before. You see the Z line, the M line. You can see how it's nicely organized. This is a myofibril, right? Or a myofilament, I'm sorry, myofilament, another myofilament, and another one. And with time, this is what it looks like. And you see, there's the Z line, and you see this fracturing, and you can look at it, and you have the slides, so you can look at it up close. You see that it's like these proteins are pulling a part of the Z line. The Z lines are starting to degrade. So we see that all the time with aging. So the question is, what causes that to happen? And when at the current time, the main mode of action would be the calpain system. Calpains are small proteins, because they're enzymes that are in the sarcoplasm. They were first identified in 1969, and they're in the sarcoplasm. They're actually around the transmembrane plaque, and calpains, I don't know if you remember that picture from clear back, in ultrastructure, it showed calpains helping some of the in some of the signaling, so they perform a lot of functions. But in general, calpain's function is to help to repair proteins. They are involved in protein degradation. And, you know, we always talk about protein synthesis and protein degradation. Synthesis, making new proteins, degradation is degrading proteins so that they can be replaced in living tissue. That total process is called protein turnover. So we know that they are very important in that process, that they are in the sarcoplasm or cytoplasm of the cell, and they are in your cells too, and they're in every cell in your body, because they help to repair. And they're an important part of the living physiology of that muscle or of that cell. They're very high in brain cells, they're very high in blood cells, and they're very high in lung cells. But in your muscle, we also find them in muscle. And so what we find, I'm going to kind of insert some more information in here, is that what they do is they actually clip subunits of the cell. So like for myosin or actin or for nebulin or titin, they don't go in and break it down like into amino acids, but it clips out subunits and or portions, and then those portions go into the lysosome where they're broken down into amino acids. So that's a very general description of how proteins are then broken down, so that then those amino acids can be put back together to form new proteins. And proteins have to have some type of turnover because we have to repair proteins. They aren't make a protein when you're 0 days old and then have that protein survive all the time. There's a turnover process, right? Well, there are actually two different forms. They're called mu-calpain and m-calpain. And I gave you in your papers some of the older papers, and the Kamari paper talks a little bit, and the Gall paper does as well. So I gave you the Kamari and Giesing, and then the Gall paper. I had to take some papers out. Aren't you glad? I love, this is probably the most papers you have in any topic. But the Gall paper kind of talks about the history of calpains, and so does the Kamari-Giesing paper. And so originally they were called CDP1, which is calcium-dependent protease 1 and calcium-dependent protease 2. So what does that mean? That means that there are proteases in the cytosol that were activated with calcium. So and then they named them calpain. It was a big deal because they're similar in structure to papain, but they're found in animal systems. They call them calpain. The mu stands for a small amount of calcium required to activate that calpain, and M-calpain, or CDP2 in the old literature, requires a millimolar instead of a micromolar. This is micromolar and millimolar. So CDP2s require more calcium to activate them. But when you look at mu-calpain is the one that's thought to be most involved post-mortem in resolution. Because initially, and I didn't give you the Calkins and Kamari papers from '88, and I had this conversation, remember when I had was gone to Lincoln, Nebraska. See, we still talk about these things. Isn't it amazing? It's 2014. And we talked about how in 1988 at RMC, Kamari and Calkins, who's another author that I gave you here, had a big discussion. And Calkins did his dissertation research here looking at lysosomal enzymes. We know the lysosome is the garbage disposal of the cell. It has a low pH, about 5.6, and its job is to take protein subunits and break them into amino acids. Then it spits the amino acids out into the cytosol so they can be used to rebuild protein. That's what the lysosome is. And what Chris looked at was whether electrical stimulation was sufficient enough to lyse the membrane of the lysosome. And the lysosome has a membrane. And so some of the enzymes involved in the lysosome are, we called membrane-bound enzymes, and these are the cathepsins B, H, D, and L, which I get on another slide here in a minute. And they go in and they break it down. They're very caustic enzymes. Because anything that can break a protein into amino acids, it's pretty tough, right? So it's in the lysosome, so it isn't just working hairy-carry. And it was always thought up to that time that the lysosomes were responsible for some of this degradation. Because some of those lysosomes, especially B, is membrane-bound and is somewhat exposed to the cytosol. So everybody thought, well, that's the enzyme system involved because that's what degrades proteins. And then we started looking at calpains, but everybody said, but you know what? It can't be calpain, which needs just a little bit of calcium to be active, right? And we know that post-mortem we get some calcium, right, out in the cytosol because of contraction, right? Permanent contraction, no relaxation. There's enough calcium to activate calpain. Probably not enough calcium to activate calpain, but enough to activate calpain. But you know what? Calpain is optimally active at pH 7.0 and at 98 degrees Fahrenheit. So it's like, well, that's when it's optimally active. Does it have any activity post-mortem and post-mortem physiological conditions? So that's the first thing, and I'm giving you some logic here so that you can kind of understand how these things evolve. So Kamari, published in 1986, he found that calpain retained about 25%, I always say about 25%, of its activity at physiological conditions in postmortem muscle, which were 5.5 to 5.8 pH and 5 degrees C. So while calpain, it isn't optimally active, it still has 25% of its activity. So it can still do some stuff, right? So that's good. That says that yes, this can be active in postmortem pHs. It's probably not a racehorse. We probably slowed it down a lot, and we have. But that activity was sufficient enough to reproduce those changes. And if you put that in a system, in a petri dish, and look at that, you can see, and you change pHs, you can see that it can still degrade. Also, calpain was found to decrease in concentration post-mortem. So if you measure at, I'm just going to put 0, 14, and 28 up on the board. If you measure calpain 0 at 0 days, you have a high level. And then at 14 days, we see a very low level. So it goes away. Oh, it goes away. How can it be active? The calpain system, including its inhibitor, calpastatin, auto-lyse themselves. So basically, if they're active, they eat themselves up. So if you see a decrease in level, it means they have been active. So, calpain decreases post-mortem. Calpain does not. So it's indicating that, calpain is the one that's active. And at least that it's active. And actually, most of its activity is in the first 48 to 72 hours. So it's active very early on, and we see it decline very rapidly. So we know that that initial levels of calpain are there, and that they also work at the Z line. Calpain was found at the Z line. There's a paper. I didn't give it to you because I could give you so many papers on this that because I love this area, right? This is good, kind of a fun area. My roommate at Clay Center actually is the one who found the first calpain at Z lines with immunofluorescence. So about 9:30 at night, I went and looked in the light microscope, and there were little green dots fluorescing. And there's a little Z line. And she's like, my protein is there. Look at where it is. And later published that, and that was ended up being a classic paper. I'm like, I got to see it second. And then we went and had a beer after. So anyway, celebrate. So we don't think that M-cal pain is really contributing. While it's maximally active at 7.5, it has little activity at 5.5. So it's a little bit more sensitive to pH changes than mu. And so at 25 degrees C is the temperature that it's also maximally active. And that the amount of calcium needed to activate it is really high. And we rarely see that level of calcium, free calcium, to activate it. And we don't really see that M-cal pain is really having a role. So it is thought that most of the changes post-mortem, that the calcium-dependent proteases are most likely responsible for those. But there's another part of this picture. There is within muscle an inhibitor, and in the old literature it's called inhibitor, CDP inhibitor, and it's now called calpostatin. And calpostatin is important because if we didn't have some way to regulate calpains, that they were just out there running amok, doing whatever they wanted to. You know, it's like turning my dog into, into a chicken yard, right? And so if he's going to degrade the chickens, this kid would like to eat every chicken, right? But maybe I'm just using him to control. His job is to weed them out and keep them from being too, to get the ones that aren't. So I have to control him in some way. Otherwise he just runs amok. And that's what calpostatin does. It's the regulator. And so it inhibits calpains. And one of the interesting things that we know about calpostatin is that calpostatin levels vary across animals. And there are actually, and I may be lying, but as far as there are 3 different isoforms of calpostatin. I'm trying to remember if there's a 4th one. I haven't read up on it lately. But anyway, so there's some different forms of calpostatin. But it also varies in level. But the other, initially, but the one thing that we see, calpostatin is like calpain. It autolyses itself when it's active. So we see a high level, and then we see it decrease with time. So the 2 parts of this 3-part system that we know influences, or that changes, we know that mu calpain and calpostatin must be involved. And so, because they decrease in level with time. So calpostatin regulates whether mu calpain can function. So calpostatin keeps it from happening, and then it autolyses itself. So what you do is you basically finally get rid of calpostatin, and calpain can work somewhat freely. Remember, by then we're at pHs of 5.6 to 5.8 and 2 degrees C, so it's not maximally active. So I want you to know about the cathepsins, B, D, L, and H. Those are the ones that are found in the lysosome of the cell. And the Gall article will talk a little bit about lysosomes. Right? And then and also the Sullivan, no, not Sullivan, the Gall paper we'll talk about a little bit, and the Kamari and Giesig mention it just a little bit. So what we find is that we know that cathepsins, they degrade actinomycin. But, actinomycin don't get degraded, do they? It's titin, nebulin, desmin, troponin T, and Z lines. So they degrade actomyosin, and if actomyosin doesn't get degraded, they must not be doing anything. And it's pretty logical, isn't it? So they are active at postmortem pHs, but they've never been shown to be released from the lysosome. They aren't, we don't see the lysosomal membrane. I mean, eventually it does, but by then the meat's spoiled, we're not going to eat it, right? But the lysosomal membrane basically stays intact through the aging process. And we don't see these enzymes being freed into the cytosol or the sarcoplasm of the cell. So, and what they do doesn't seem to happen. So it pretty much eliminates them from activity, doesn't it? So these are just some things that I pulled up about calpains and calpastatins. Don't worry, I'm not going to ask you to give me the sequence and to understand the protein. These are fairly small proteins that are enzyme either mu or M. There are some membrane binding potential. That's why they are involved in with interferon and some of the signaling of the cell. Makes sense, they're involved in degradation, they should probably be involved there. We know that cells contain calpains. They're for general housekeeping. They're the cleaner upper out in the cytosol, all right? So they help with fusion and differentiation. They help with membrane protein degradation. They help to activate protein kinase C isoforms. They help degrade the membrane-bound hormone receptors and focal adhesion complexes. We haven't gotten into all that, but it's related to interferon. And then they help with skeletal protein remodeling. I'm not going to ask you to repeat that list to me. But what I want you to understand is that calpains aren't, let me rephrase this. If you were a graduate student and only going to RMC and not doing very much literature search on calpains through the 80s and 90s and early 2000s, you would think all calpain does is degrade the Z-lines, titin, nebulin, and desmin. And I just want you to know it's a very active component within the cell and how that cell functions. And it has multiple purposes, all right? Oh, and there are 3 polymorphisms. So calpastatin helps to regulate calpains, and this slide is just, I'm not going to ask you, again, to know that. I want you to see that, there's calcium is involved in this, and here's calpain, and there's calpastatin, and that calpastatin helps, allows calpains to be active. There's a lot of information on how that happens. It's very important within the physiology of that cell. And I want to plant that thought. And if you're interested in it, I can help direct you to some of that information. But calpastatin regulates calpain. That's the point I'm trying to get. And then I made this very simple slide because I have to teach this to beef producers. So, you know, if I put some of these other things up there, they'd be gone, right? They'd be thinking about, I wonder what hay prices and corn prices are doing today. And they'd be on their phones checking things, right? So I put this slide together to kind of show what's going on. Okay, you should recognize this matrix down there. That's the sarcomere, the myofilament of the muscle fiber. And this policeman represents calpastatin. And this shark represents mu calpain. And so initially post-mortem, there's a lot of policemen. But if you have a bunch of policemen out in the water and a bunch of sharks, who's eventually going to win out? Because the policemen don't have guns. The sharks, right. So what happens is when the policeman's active, then he dies. When the shark is active, then it dies too. So eventually you get all of them gone. But in the meantime, the shark gets to come in and break the Z-line. There's titin. I don't have desmin in here, but it starts clipping away at all the things. So that kind of helps put that in perspective. Kind of a fun one to look at. So we do know that calpains control focal adhesions, assembly of many tissues. It's involved in lots of repair mechanisms, and that it's been shown to degrade troponin T, also filament, desmin, nebulin, and titin. I only gave you the other one, troponin T, desmin, nebulin, and titin. I'm not going to ask you about filament, all right? And that we know that calpastatin activity differs across muscles. And this is the longissimus muscle. And look at, this is how much calpain activity, or calpastatin activity it has, versus the semitendinosus, one of the toughest muscles. And so these data were used, and there's a number of other data as well. And here's a fragment of total desmin is in the gray. And notice that the more calpastatin you have, the less desmin you have, indicating that calpastatin is degrading desmin. There's a very high relationship. You can look this paper up if you want to. But I put that up there just to show you, to reiterate the fact that there is a relationship between amount of calpastatin and how much of the structural proteins are there. And that more calpastatin is present across different muscles, and the more tender muscles tend to have less calpastatin, the tougher muscles, more calpastatin. And that makes sense, because those tougher muscles are high-use muscles, aren't they? So they're going to need more protein turnover. If Biscuit and I are now walking, because they told me he needs to lose a pound. I'll show you guys all a picture of Biscuit. He's a dachshund. So, but he thinks he's a border collie, so we're not telling him anything different. And I know he could have won the wiener races this weekend if I would have taken it, because he runs like a border collie. Anyway, so, that's just a side. So, you know, we're out there walking and everything, and the muscles that are working the hardest are my leg muscles, aren't they? And so they're not used to working. They're the ones that are going to have more repair. They need more calpastatin. They need to be able to function, right? So, and we know that even as we restrict feed and have animals grow, that there is a change in level of calpastatin and regulation of, and how much active mu calpains. Oh, wow. All of that tells us that the level of calpastatin and how active then mu calpain can be most likely explains a lot of the changes. Now, I use the word most likely because it doesn't explain all of the changes. There's a lot more going on. And luckily, we don't have enough time in this class to talk about all that. But the basic principle is with calpastatin. And I love these pictures because I just can't resist. I'm sorry. But we talked about focal adhesions. And what I just want you to see, there's some things that you should recognize. Don't worry, I'm not going to have you know this on exam. But again, it's to kind of help let you sit down and see, kind of convince you, I guess, that calpains are involved. This is just showing how there's some beta-1, interferon, some talin, there's F-actin. Oh, F-actin, right? We know what that is, right? Yeah. And notice that with calpain, we see that its action is to break things apart. And this is in the focal adhesion breakdown. So, calpains are very much involved in breakdown. So, all that, right? I hope I've convinced you. I hope I've convinced you that calpains are involved. You know a little bit about calpain. So let's talk about from more of a practical standpoint. And I put all those other things in. Partly, I don't mean to have it panic you. The last two slides, I'm not going to ask you a question over it. But I want you to understand that this is a lot more complex than what I've shown you, right? And it isn't only just working here, but it's involved, calpains are involved in lots of things within that cell. So, I promised you some data on a big study. So this is a summary of a 5-year study where we had 16 different sires, Bos indicus sires. One of them was a Navori sire, the other ones were Brahmin. And for those of you who are poultry people, within the beef animals, we know that Bos indicus animals tend to be tougher than the Bos taurus. And Angus is our main Bos taurus breed. So we had an Angus sire. And then we produced, on Hereford and Angus cows, we used these Bos indicus cattle, or sires. And they represented the majority of sires used in the U.S. The big sires. American Bronc Breeders Association supported this work. So what we did is over 5 years, we bred cows, and we took the steers from those cows, progeny from those cows, and we fed them out, and then we harvested them, and then I took the meat and I graded the carcasses, and I took a steak randomly, except for 14 days. They only paid me to do the 14-day steak. I had to buy the whole strip loin. I'm like, I have all this meat, I'm going to do a lot more with it. So at 1, 7, 14, 21, 28, 35 days, I did shear force. Now shear force is a mechanical measurement of tenderness. And so a higher value means that they're tougher. And there's lots of animals in this, but I condensed it down into the red line and the blue line. And I don't think I've given you, maybe later on I might give you all of the sires, but it's a very colorful slide. And one of the things that, and we measured calpains and we measured calpastatin on these animals, right? And so each one of these dots represents a lot of animals over here, and I did all the things you're supposed to do with counting for year effects and all that thing, which you don't really care about, but I am. So I did this with Dr. Andrews. So look at what happens in day 1. We find that these are the Bos indicus, the cattle that are tougher. They were tougher than the Angus on day 1. And when we looked at calpastatin, these animals had more calpastatin. But with time, calpastatin decreased. And this box represents the same shear force. And the reason the 14-day is lower, I can tell you guys this. I have a different slide I show producers where I took that out because it confused them. I only got paid to do the first strip loin steak, and I had to do the first strip loin steak, and it's always more tender than the other ones for location because there's a 13th rib there, all right? So that's an artifact. Why it doesn't dip down because it would dip down. The other ones are randomly assigned by location except for 14 days. That's why you have to randomly do it. But, that's what I was paid to do, so I had to do it that way. But what I'm trying to do here is get resolution of rigor, right? Get meat aging. Get some, I want to weaken this actin-myosin relationship. I want to get Titin, Nebulin, and Desmond degraded. I don't really care that much about troponin T because it's a regulatory protein, right? But it's going to degrade too. But Titin, Nebulin, and Desmond, boy, those are the big ones, and then degrade the Z line. Boy, if I can get those 4 things done, I'm going to get more tender, aren't I? Because the house is falling apart. And notice that by 7 days, these animals that have less calpastatin, I would write on here, I wonder if I can write on here today. Nope. Maria Kelly, I need you to push it back. Sorry. So they had less calpastatin, so they had less policemen. The calpain started working. I did sarcomere link. I did all kinds of things with these animals. And these had more calpastatin, and it took them 14 days. But they eventually got into the box, didn't they? And they eventually degraded, and I had pictures of Z lines and all kinds of things, and fragmentation index and all kinds of stuff, right? But it took them longer because they had higher calpastatin to begin with. And so most of the effect of resolution of rigor occurs in the first 14 days. This graph doesn't really show changes. You will continue to get some changes up to 35 days. We're going to talk about that in a minute. But 14 days removes the variation associated with calpastatin. And some animals will never age, and that's not a calostatin issue, we'll talk about that. But if they have, if what's keeping them from aging is